Hello everyone, and welcome. Today, we're going to explore a powerful and unique laboratory technique called hydroxyapatite chromatography. It might sound a bit complicated, but the core idea is actually very clever and useful for separating biological molecules. First, let's break down the name. Hydroxyapatite is a mineral form of calcium phosphate. It's the very same substance that makes our bones and teeth strong and hard. In the lab, scientists create tiny, porous crystals of this material and pack them into a tube, which we call a column. This column filled with hydroxyapatite crystals is the heart of the separation process. So, how does it work? This is where it gets special. Hydroxyapatite chromatography is known as a mixed-mode chromatography. This simply means it uses more than one method of interaction to separate molecules at the same time. Let's look at these two modes. Number 1 is cation exchange. The hydroxyapatite has negatively charged phosphate groups on its surface. These act like tiny magnets for positively charged molecules, such as basic proteins, binding them to the column. Number 2 is calcium metal affinity. The hydroxyapatite also has positively charged calcium ions. These, in turn, attract and bind to negatively charged molecules, like acidic proteins or the phosphate backbone of DNA and RNA. So, this single column has both positive and negative interaction sites, giving it a unique ability to grab onto a wide variety of molecules with different strengths. Imagine you pour a complex mixture of proteins and nucleic acids through the column. Some molecules will bind very tightly, some will bind loosely, and others might not bind at all and just wash straight through. To get the bound molecules off the column in a controlled way, we wash the column with a buffer that has an increasing concentration of phosphate. These new phosphate ions compete with our sample molecules for the binding sites, effectively pushing them off the column. Molecules that were weakly bound come off first, and the most tightly bound molecules come off last. By collecting the liquid that comes out in separate tubes over time, we can successfully isolate and purify the different components of our original mixture. This technique is incredibly valuable for purifying antibodies, enzymes, and nucleic acids, especially when other chromatography methods fail. And that's a basic look at hydroxyapatite chromatography, a versatile tool for scientists. Thank you so much for watching. If you found this explanation helpful and want to learn more about science and lab techniques, please consider liking this video and subscribing to the channel. Your support truly helps in creating more content like this. See you in the next video.